we'll be talking about neuropsychology of memory today. We're going to talk about the history of this area of research, which gives us a little bit of insight into how we talk about memory and memory formation in particular, and memory consolidation. We'll take a look at some pretty famous case studies and talk about what we can learn from them. So we'll start with kind of this historical perspective in what we refer to as the search for the Engram. This is essentially how there was this very long, not particularly fruitful search to find where memory was stored in the brain. Turns out memories are sort of stored everywhere in the brain. We'll then talk about patient HM. HM was mentioned sort of briefly in the Memory Hackers video. So we can take a look at his case and what we can learn about memory from his case. One of the most widely studied individuals, in fact, over 50 years of research on patient HM from his surgery until his death in about, 2009, so not that long ago. So pretty much a solid, almost 60 years of research about patient HM. So from there, we'll talk about amnesia patients and models of human memory, and talk about the neural basis of memory, and then finally finish up with some emerging frontiers in this area. So we'll start with this search for the Engram. Carl Lashley is the sort of first historical figure in this area. He believed that there was a single biological locus for memory. So he set about trying to find out where in the brain memories were stored, memories were kept, what was the critical area for memory formation. And he was kind of right in that there is a single biological locus that's critical for memory, but it's not where memories are kept. Rather, it's where memories are formed. Many rats lost their lives to this fruitless search for the location for memories in the brain. So what Lashley would do is he would train a rat in a maze task, conduct some stereotactic surgery, removing part of the rat's brain, put it back in the maze. Rat could still find its way through the maze. So Lashley was largely unsuccessful in his search for where memory was located in the brain. So he couldn't figure out where memories were stored in the brain. While he had the right idea, he was going about this in the wrong way. As it turns out, memories are stored throughout the brain, but there is a part of the brain that's critical for their formation. So then his student, Donald Hebb, espoused a theory based on connections between neurons as being the critical way in which memory works. We actually now refer to this idea of connectionists, or connections between neurons, as an important part of learning as Hebbian learning. So it sort of forms this basis of many neural network models. So this idea that the way neurons are connected is an important part of memory formation. And of course, we saw Eric Kandel's work in the Memory Hackers video, where we do start to see how neurons are connected to one another is an important part of memory formation. So Lashley and Hebb start out with some very important ideas. And in fact, they really form the basis for our modern understanding of the neuroscience and neuropsychology of memory. It turns out they were both right in the wrong ways. So they had the right ideas, just simply hadn't taken their research exactly in the right direction. So their lasting contributions are this idea that there is a localization of memory functioning. We now know, of course, that the hippocampus is responsible for the creation of memories. It's not where memories are stored, but it's how memories are formed. And that's where Lashley sort of made his mistake. He never actually tried lesioning the rats before they were trained to see if he could affect their encoding. He was trying to find out where memories were stored. So we do know there's this critical area for memory, and that's, of course, the hippocampus. We also know that connections between neurons are an important mechanism for learning through long-term potentiation, synaptic connections, synaptic branching, increased connections between neurons. There's all sorts of different ways, but this connection, strengthening of connections between neurons is an important part of the learning mechanism. So Lashley and Hebb were both correct in terms of how the fundamentals of how memory work. They just didn't get all the details exactly right. So we're going to talk about long-term potentiation and its role in learning later on today. So that takes us then to patient HM, which is Henry Mollison. It's weird. For most of my career, we didn't know who patient HM was. We certainly didn't have pictures of him. He was just this mysterious person that we talked about. Had his temporal lobe surgery in 1953, where they removed pretty significant portions of his left and right hippocampi and his left and right temporal lobes. You can see this is the normal brain here, and these are the parts of the brain that were excised. So fairly dramatic neurosurgery. Now, HM had terrible temporal lobe epilepsy. And had he not had some sort of surgical intervention, it's likely that HM would not have survived very long. So this, in all likelihood, was a life-saving procedure. Unfortunately, is also a dramatically life-altering procedure. So here we have, it also caused some pretty progressive sort of brain loss over time. So this is an MRI of HM in 1992. We can kind of see losses, and then you can also see his brain really starts to shrink when we get out to 2003. This is, of course, 50 years after his surgery. He lived about another 16 years after that. So pretty long life, considering. So he has this temporal lobe surgery. After the surgery, they discover that he has incredibly dense anterograde amnesia. After his surgery, HM really formed no new long-term episodic memories. So he didn't remember anything that happened after his surgery. He still remembered where he grew up, where he lived. He remembered all of his past prior to the surgery, but nothing really after that surgery in 1953. But, and we saw a little bit of this in the Memory Hackers video, he was still able to learn. And this is really the most remarkable part of HM's story, is he could still learn how to do new things. So his intact learning included things like mirror tracing, and this is the thing they talked about in that Memory Hackers video. He got much better at this over time, though he never had any memory of having done so. He could also do this pursuit rotor task. I'm bound and determined to find one of these. There's got to be one somewhere. This is an old school task. They actually used it as sort of an I.O. task in the old days. But essentially what you had to do in this pursuit rotor task is chase this little metal disc around this turntable with this metal arm, and then it would record how long you kept contact between the arm and the disc. And so it was basically a motor skills task. It sounds easier than it really is. Having actually done this, when I was an undergraduate, we had a measurement and testing lab that we took, and I walked in late and rather hungover one day, and ended up having to do this task in front of the entire class. And there's nothing more difficult than chasing something around a rotating turntable when you're not feeling your best. So it's actually pretty difficult to do, but HM could learn how to do it. You can also learn how to do this Tower of London. Sometimes it's also called the Tower of Hanoi. If you're interested in doing this puzzle, you can actually download an app. It's kind of a boring puzzle to do. But essentially, this is the apparatus. You have 3 pegs with a number of decreasing in size rings. The goal is to move all of the rings to the 3rd peg. You have to move 1 ring at a time to another peg, and a larger ring can never be on top of a smaller ring. Those are all the rules of the game. And H&M could learn how to do this in the fewest moves possible, but again, without any memory having done so. So he didn't remember any of this, but he was able to acquire these new skills without any actual memory for it. There's also some evidence that H.M. picked up sort of new semantic knowledge. That is, he could, he sort of knew who Richard Nixon was. He read the paper every day. And so he sort of knew some things about modern times, but not in the way that you or I might. So he did actually acquire knowledge, but not at the same rate that any of the rest of us might. So over time, he was clearly learning, but not having any conscious memory of having done so. So it's really remarkable. What this establishes is that there are conscious and unconscious forms of memory. That is, he's able to learn all of this without any conscious access to the past. And that's a pretty groundbreaking finding. So what H&M and others taught us about memory is that short-term memory and long-term memory are biologically distinct. H.M. had perfectly intact working memory. So we could do digit span tasks and all that sort of thing. We certainly know that some long-term memories are stored throughout the brain because he still had access to his past. And of course, the hippocampus is necessary for the creation of new memories. And this is a critical part of the brain that Carl Lashley didn't find because he was looking for where memories were stored, not where they were created. It didn't occur to him that there was a critical part of the brain for memory creation. So I don't think it's an overstatement to say that H.M.'s case completely, radically altered our understanding of the human brain and certainly our understanding of how memory functions. We also know, based on H.M.'s work, they work with patient H.M., that there is more than one type of long-term memory because H.M. could learn things that he couldn't remember. So this is what we call a dissociation. That is, we can dissociate learning from remembering because the two are distinct from one another. Your book talks a lot about dissociations in this area. So we get a lot of important information from this and other case studies. So there are other patients like patient KC and others. H.M. certainly, I think, is the one that radically changed our understanding. So I want you to watch this clip about Clive Waring. Really remarkable study. Really remarkable case. If I can get it to play. There we go.
One man is consigned to live entirely within the present, with terrible consequences. Clive Waring has the worst case of amnesia ever known. 20 years ago, he lost his memory, and now his wife, Deborah, is the only person he recognizes.
Really only has less than 30 seconds memory, and sometimes it's as little as perhaps 7 seconds. It's as little as a sentence.
I'm going to see your sister, Adele. My daughter's got married recently.
Oh, is she?
Indeed.
Oh.
And so we're here in the party.
Funny how the ladies apply different stuff when they get married.
Do you know who I'm going to see tomorrow?
Buckingham.
No, really don't.
Oh.
You don't know.
Oh, I see.
Do you know why I'm going?
No.
She's having a party at my house tomorrow.
She works for you.
No.
No.
Do you know why?
No.
It's to do with her daughter.
I see.
Do you know why her daughter's having a party?
No.
Guess.
No, I don't.
feeling.
Oh, I see.
The sentence he is in, he will probably have forgotten the sentence before. You ask him a question, and he'll give you an answer, but while he's giving you the answer, he's already forgotten the question. That's how short it is.
When you see your kids tomorrow,
You see my kids?
yeah, you're talking.
What are they up to now?
If you know what they're up to,
Mm-hmm.
guess what you think they might be.
I know what their A-level was. They haven't got their A-level last time I came. So there's more possibility they do, yeah. And that's who you could possibly guess.
What? Where do you think they are?
I know. There they are.
There's Aaron, his boy, behind him.
Look at that dog. I've never seen a human being before. Never had a dream or thought. The brain has been totally inactive. Day and night the same. No thoughts at all. As far as I'm concerned, the doctors have been totally incompetent. I've never seen a doctor the whole time. Oh, look!
All right, so that's Clive and his wife. There's really a lot. You can find sort of more videos. There's a much longer version of that video from the BBC. What particularly stood out to anyone? A couple things that I think are particularly important from that clip. Anyone? Yeah. It's a really unusual case. He had a really bad case of viral encephalitis, and if he'd have been a few years earlier, he probably would have died. A few years later, he probably would have been fine. But it attacked his entire brain, and so it's a really unusual case. You don't usually get this kind of very complete loss of memory. So that's certainly one part of it. It's really unusual. In fact, it's one of the only ones I know about. Yeah, he was a conductor for the BBC. And so we're sort of a very famous musician in the 1980s, 70s and 80s. And in fact, if you sit him down at the piano, he'll say he doesn't know how to play, but then he actually can play. So that's exactly it. One other thing. What happened when he saw his wife? Yeah. Right. But that emotional memory is so incredibly intense. And that's one of the things that's really remarkable. He knows he's got kids. The knowledge about his life is still there, right? So while the episodes are gone, his knowledge about his life is still mostly intact, right? So one of the things we saw in thinking about autobiographical memory is that it's both semantic and episodic. And so he sort of has all the knowledge about his life, and he's got bits and pieces of his past. But that emotional memory is clearly intact. So I think it tells us that emotional memory is a little bit different. Yeah. Yeah, this is one of the things that gets well, I mean, it's been more than 20 years. So, yeah, it's really remarkable that he's survived this long. Obviously, he was in a long-term care facility because as soon as he walked out the door, he wouldn't know where he was. But my guess is his knowledge of who she is will sort of update, I guess. I mean, it's hard to say exactly. There's some cases of people with prosopagnosia, so they can't recognize faces, but as soon as the person talks, they can recognize them, and they say it's like their face all of a sudden comes into focus. So their knowledge about the person helps them recognize it. Yeah. Well, you have to think about it. I don't know how old his kids were, but once you're past 20, he looks the same enough, right? But when you go from 8 to 25, that might be a little bit more difficult. So I've never seen any interactions with him and his kids, so I don't have a good answer. If you watch the whole video, I don't know, she might talk about it in her book too. So I haven't watched that whole series in a while. So I think that could be part of it. I mean, he's expecting his kids to be. I think that might be part of it. But I don't know. It's a really good question. Really, I mean, it's tragic, very tragic, but also, I mean, it's a really fascinating case to me, particularly how much infection he has for his wife after all this time. I mean, that's got to be incredibly hard for her, I would imagine. So, anyway, it's a really remarkable case that tells us a lot about emotion and memory and skills and memory, that they're different from our day-to-day experiences, and so our day-to-day life episodes are a different beast. So I want to talk a little bit about amnesia patients. I've talked about the extent of memory deficits we typically see, introduce some memory systems. Some of that I'll be able to skip through. Some of this lecture was designed as a standalone. Actually, I think this is the lecture I gave when I got this job. Talk about the anatomy of amnesia and then the temporal nature of amnesia. So the first thing to understand about most people with amnesia is their explicit memory losses tend to be very global. Particularly if it's anterograde amnesia, they just have no conscious recollection following their injury. So if we bring them in to do like laboratory studies, they can't do things like free recall, cued recall, recognition task. It doesn't matter if we read it, show it to them, have them imagine it. It doesn't matter. They simply have no functioning explicit memory. They tend to have spared working memory and spared skill learning. So usually learn new skills, can't often use them because they don't remember they have them. They tend to have spared implicit memory too. This is where we start getting a little bit more into formal studies of memory. So we're going to take a look at sort of one of the classic studies in this area where they took a few amnesia patients and studied their memory versus sort of normal patients, normal people like yourselves. In some of my work, we've done similar studies, but we actually induce amnesia with a drug called midazolam in healthy patients. So it's a little bit easier to do. Amnesia patients are hard to find. It's much easier to just temporarily create them. And it's very temporary amnesia. Many of you have actually had this same procedure done. If you've had your wisdom teeth taken out, if you've had any sort of outpatient surgery, oftentimes they'll give you Versed or midazolam so that you don't remember the procedure, even though you're awake for the procedure. If you've ever had an endoscopy, you've for sure had midazolam. So things you have to be awake for that they don't want you to remember the unpleasantness involved. They'll give you this drug so you don't remember any of it. So in this particular case, classic study by Graf, Squire, and Mandler. Control patients much better than amnesia patients in pre-recall and cued recall. But this implicit task, and this is what we call an indirect test of memory. These are tests in which you don't actually have to consciously recollect what happened. And in a word stem completion task, you're just asked to complete the word with the first word that comes to mind. It's counted as correct when you complete it with a word that was presented on the study list. And in fact, the amnesic patients are more likely than control patients to complete using a word from the study list. So they don't have to consciously access that memory. So they're actually as good, if not better than, sort of quote-unquote normal patients. So this is where we kind of are in this large area of memory. Amnesia patients in particular, this events part of their memory is what's particularly gone. Learning of new facts becomes degraded because learning new facts relies on episodic memory. They can learn new facts eventually, And all of this is spared. So we think that word stem completion is probably somewhere in this perceptual representation system or sort of non-associative learning. That's how these participants are able to access those memories. We'll talk a little bit about these implicit connections not having reciprocal connections back to the cortex, which is why they have no conscious component. So, of course, episodic memories, semantic memories, we've been through this already. So no need to write all of this down. Implicit memory, of course, includes motor learning and perceptual learning. The anatomy of amnesia generally almost always involves, particularly for anterograde amnesia, the hippocampus. This little chunk of the brain here. In particular, I want to talk about the dentate gyrus and the CA subfields of what's called Ammon's horn. So now we're getting down into the much greater detail of starting to understand how the hippocampus is involved in this creation of memories. So this part of the hippocampus has 4 layers we'll talk about. So we go from the dentate gyrus up into this area called Ammon's horn. So the dentate gyrus includes what are called granule cells. This is the most important part of understanding the dentate gyrus is this is where new neurons are created by the hippocampus. This is at the cutting edge of neuroscience, is understanding this process of neurogenesis. 20 years ago, we didn't know that new neurons were created in the brain. We thought you were born with every neuron you ever had. Turns out that's completely wrong. And this is one of the areas of the brain that's very involved in creation of new neurons. So this is happening at the dentate gyrus, and we think this is very important for formation of memories over time. And this is happening again in these granule cells in the dentate gyrus. This part of the brain is particularly susceptible to anoxia, so any loss of oxygen, and also to elevated cortisol levels. Cortisol, of course, is released during long-term stress experiences. It's also elevated in depression patients. We're going to talk about this when we get to the end, but we think these are tied together with depression somehow. So we're going to talk a little bit about that here in a bit. So the dentate gyrus is an important part of our memories, particularly, we think, for structuring of permanent long-term memories and with forgetting. So when we get into the CA subfields of Ammon's horn, the first and third subfields are made up of pyramidal cells, important cells in the hippocampus. These are also very susceptible to injury, particularly the CA1 subfield. So again, loss of oxygen, carbon monoxide poisoning, toxins, that sort of thing, can be particularly damaging to the hippocampus. Particularly, again, when we start talking about things like cortisol. There's a disease called Cushing syndrome, in which primarily women have tumors of the hypothalamus that cause extremely high levels of cortisol to be released, and as a result, they tend to see pretty striking damage to the hippocampus. That once the tumor is removed, it's resolved over time. One of my, I guess now we're not related anymore since my mom and her son divorced, but it was my mom's husband's mother, actually had Cushing syndrome, and they went in through her nose to remove the tumor. Pretty remarkable, because your hypothalamus is basically right here. Very hard to get to any other way. So they go in, drill up through the sinuses, and remove the tumor that way. Kind of remarkable, really. Again, these CA1 and CA3 subfields are particularly important for memory formation. We'll take a look at kind of a model of how this all works together here in just a little bit, but this is an important part of also understanding how memories are formed over time. this all interacts with what we call the medial temporal lobe complex, which is the MTL. So it's just simply tucked in underneath the temporal lobe, connected with the hippocampus and its associated areas. The three primary areas we're going to talk about are the entorhinal cortex, the pararhinal cortex, and the parahippocampal complex. This is where we start to see memories being sort of permanently stored as they move up through these cortical areas. So these are three important pieces of this MTL complex. There are some subcortical bits and pieces, for want of a better term, that help put together this all into important memory systems. That is the technical term, by the way, is bits and pieces. We'll take a look at these subcortical areas at the diencephalic midline. So the mammillary bodies are an important part of memory formation. You can see in this top, it's kind of hard to see, these sort of healthy control mammillary bodies, and then these have severe mammillary body atrophy. This is particularly an important part of Korsakoff's amnesia. Korsakoff's amnesia is caused by long-term alcohol abuse. Essentially what happens is long-term alcohol abuse causes thiamine deficiency because your liver is not able to produce thiamine or process thiamine. And as a result, then the mammillary bodies can suffer. There's also one very unusual case of a person who was fencing without, they were just screwing around in the guy's office, I think is the story. But the guy was fencing without a mask, and the fencing foil went in his nose and pierced his mammillary bodies and had instant amnesia. So, try not to fence without a mask. Important part of that. By the way, if I haven't talked about, have I told you guys one of my favorite shows on TV that there is called I Was Impaled? Oh, it's the best show ever. It's all about people, it's all real cases of people that get impaled on random stuff. So, like, this woman falls off a ladder in her yard and lands on a shepherd's hook. You know what that is? It's essentially a metal hook that you hang a plant on. So she lands on the hook and it goes into her eye. She has to take the hook out of the ground and walk to her neighbor's house and ring the doorbell. They end up medevacing her, cutting off part of the hook. They take her into the hospital, and they just realize that the best thing for it is to pull the hook out. And it turned out all it had done was push her eye aside. So it was perfectly fine. Didn't lose any vision. Fine. I actually, one night, watched like 7 episodes in a row because I couldn't stop. But it's available online, so look into that. I don't think the guy with the fencing foil is there, but we should write in and get them to do the story about it. So then the medial dorsal thalamic nuclei and the anterior thalamus are also important parts of memory formation. So how does amnesia happen? Well, it tends to happen due to, of course, brain injury. Most commonly are strokes, tumors, aneurysms, and disease. So this is what we call organically caused amnesia. This looks to me like a glioblastoma. I'm just going to guess. I honestly don't know. I can't remember what I got this from. That might be actually an aneurysm. Aneurysm, what happens is blood vessel ruptures and it's like a bomb going off in your brain. Stroke, of course, is ischemic stroke. A blockage causes atrophy of the brain. Fortunately, we have really terrific procedures for clearing strokes compared to what we used to. When we talk about language, we'll talk about there's a mid-communicating artery stroke that often causes aphasia. The other types of somewhat natural causes are loss of oxygen, so lower levels of oxygen, which is hypoxia, or a lack of oxygen altogether, which is anoxia. Or other types of toxicity can also cause amnesia, cause damage to the brain. somewhat rare. Again, the kind of toxicity we'd be talking about is things like carbon monoxide poisoning, or carbon monoxide replaces oxygen in the blood, other issues like that. Penetrating injuries, and like the fencing foil. Not sure what happened here. That looks like an arrow to me, but you can find all sorts of weird penetrating injury X-rays online if you have time to go surfing for that. We've talked already a little bit about traumatic brain injury and closed head trauma. We're going to talk about temporally graded amnesia with closed head trauma. So this is another very common cause of amnesia. Usually this resolves over time. Oftentimes people will lose a chunk of time, but not, it usually recovers over time, or they'll just lose, time surrounding their injury, depending on the severity of the injury. This becomes kind of problematic, particularly in the time when somebody's trying to recover from this injury, because they kind of just keep losing track of what's happened. Finally, people who undergo electroconvulsive therapy also can lose chunks of time. Electroconvulsive therapy is essentially an intentional brain injury. Still done. It's actually done more than it was somewhat in the past. It's a much better procedure than it used to be, much smaller seizure. So what they do with electroconvulsive therapy is they induce a seizure in the patient by inducing a current into the brain. And this actually does seem to really help some severely depressed patients. Carrie Fisher, who unfortunately recently passed, writes about it in her autobiography. By the way, if you want one of the funniest books in the world to read, you should read her book Shockaholic. Really funny. Actually, the one before that is Wishful Drinking. It's also really funny. But she talks a lot about this kind of memory loss because of that. One of the things with ECT is, while it's unfortunate for the patient, from a scientific perspective, it's fascinating because we have a scheduled head injury. You don't tend to get a scheduled head injury, and so you can actually sort of have the patient track what happens over time, and so you can actually get pretty good, verifiable data about their memory. And so you tend to get what we call this temporally graded amnesia. So they can lose, as their brain's recovering from the ECT, a couple months following, so they get a little bit of anterograde amnesia and a little bit of retrograde amnesia that diminishes sort of over time. So they lose sort of a lot close to the ECT and a little bit less before that. This tells us a lot about what's happening in memory formation, because this disruption in brain functioning disrupts the encoding of new information, but also then the storage of previously learned information. So there's a biological process that's going on that's taking these previous experiences and making them permanent memories that gets disrupted. And that's a really important part of this. So the final way we can do studies of amnesia is doing pharmacological amnesia. So this is some of the work that I've done where we use this drug midazolam. It causes very dense, very short-term amnesia. So in a typical midazolam study, a patient would get injected, wait a few minutes, and then we had maybe 15 minutes or 20 minutes of very dense amnesia. Patient's awake, answering questions, and then we wait about an hour and a half. The drug's completely worn off, and then they just don't remember what happened in that short period of time. So this provides a really powerful technique for studying amnesia. So there's still people doing work in this area. Lynn Reeder up at Carnegie Mellon, Marian Mincer up at Johns Hopkins. Really interesting stuff. So one of the things that's really important in terms of what we can learn about amnesia is understanding this temporal nature of amnesia. So anterograde amnesia is, of course, a deficit in new learning. Patients aren't able to form any kind of new memories. whereas retrograde amnesia is this inability to access memory for events prior to the injury. Most cases of amnesia are shorter term and are associated with temporally graded amnesia. These sort of long-term permanent amnesia patients are very rare. In particular, you don't very often get retrograde amnesia patients with sort of complete loss of their lives. You don't get, this sort of what I call soap opera amnesia, where people show up, don't know who they are, or where they came from. That just doesn't happen. Very rarely, and usually when it does, it's psychogenic amnesia. It has nothing to do with a head injury. So these cases of temporally graded amnesia provides us a lot of information of this underlying process by which memories are formed. So by looking at this pattern of how memories are lost during this process, tells us a lot about the temporal nature of memory formation. So by looking at what people can remember before and after a head injury, we get a really good idea of how this is all unfolding. This is really important. This is a really important start to understanding this process. So here we have a patient with very severe head trauma. They're in a coma for 7 weeks. One of the things that is sort of a good rule of thumb is the longer somebody's unconscious from an injury, the worse their memory is likely to be. Right? So somebody that's out for an hour is going to have a little bit of memory loss. Somebody's out for a day, it's going to be worse. This person was out for 7 weeks. So we have complete and total anterograde amnesia, nothing following the event. About 2 years of retrograde amnesia, complete and total retrograde amnesia, and then just not very good memory prior to that. So this person's got a pretty bad memory. But fortunately for this patient, this resolved over time as they got better. So this is, I can't remember what the time frame is here. Sometime later, we get a few memories starting to be formed following the injury. The complete retrograde amnesia is now at 1 year. Everything's a little bit patchy for 4 years prior to that. Then finally, we get, again, sometime later, I want to say this is a year or two later, their retrograde amnesia is only about 2 weeks prior to the injury. They've lost about 3.5 months after the injury, but everything else is perfectly normal. So they've sort of lost, about 4 months of memory during this sort of time frame. So we call this shrinking retrograde amnesia. And what's happened is, as the injury resolves itself, what we think has happened is this disruption has caused a couple of things to happen. Obviously, it takes about 3 months for the hippocampus to recover, so we start getting formation of new memories. But we also think that the hippocampus is part of our memory retrieval process. And it's not very often people get both of those things damaged, but in this case, the patient is unable to retrieve sort of more recently formed memories. Everything before that is much more stably formed. And over time, what's happening is things are getting consolidated up into the cortex, and so they're less dependent on the hippocampus. And so eventually, once this patient has healed, they can retrieve all of their memories that aren't completely consolidated, and they're able to start forming new memories again. So that then leads us to look at what is this neural basis of memory. So we'll start with a brief overview, talk about consolidation in the hippocampus, talk about the cortex, and then talk about some other types of memory and how they are involved. So, the medial temporal lobe structures modulate both explicit and declarative memories. So let's kind of step through this flow diagram a little bit. So we have temporal lobe structures that are interacting with the neocortex. We have sensory and motor information coming into the neocortex and into the temporal lobe structure and into the medial thalamus. And these kind of interactions. So the temporal lobe structures are interacting with the prefrontal cortex. This is probably one of the most important parts for how we form memories. So the important parts of declarative and explicit memories are the hippocampus, the pararhinal cortex, the temporal lobes, and the frontal lobes. These are the critical pieces of the puzzle. Now, in that patient we just looked at, they probably had damage to all of these areas that took time to resolve. In Clive Waring's case, he had permanent damage to definitely the temporal lobes, the frontal lobes, and probably hippocampus and pararhinal cortex as well. One of the things that happens with encephalitis is your brain swells, and as a result, you end up damaging fairly significant portions of your brain. So this is a beginning to look at what are the important structures that we're going to talk about. The process involved here are, we know some of the things that are happening. This is an area where there is a ton of research being done in terms of how this exactly works. I would love to tell you exactly how your memories are formed, but we don't know. So if this is something that interests you or excites you, there is lots of work to be done in this area. So the first major process I want to talk about is what we call consolidation. And this is the process by which memories are strengthened to allow for long-term retention. Memories are consolidated. We think this is one of the important processes that's happening when you sleep. Sleep's particularly important for long-term memory formation. It's also a process that takes time. It's the reason why your dreams include a lot of weird stuff, because memories are consolidating from all over the map over the last several years of your life. And so as this takes a long time, and so you start catching these weird glimpses of stuff that you haven't thought about in a long time, but are parts of memories that are still being consolidated. So the temporal nature of amnesia provides evidence for this phenomenon. So memories that are lost just prior to a head injury are ones that were never consolidated. And then, of course, memories following a head injury are just simply not formed in the first place. And so that retrograde loss of memory in a head injury patient is evidence of memories that weren't consolidated. So we'll take a look back at that patient we were just looking at and take a closer look at this phenomenon and how we think it provides evidence for the idea of consolidation. So here's what I was trying to look at. So this is 5 months following the injury, 8 months, and then 16 months after the closed head injury. And so what we see then is these memories were never consolidated, whereas everything probably prior to those 2 weeks was at least partially or completely finally consolidated. It's lost here because those structures aren't able to access those memories. And then these memories following the injury are never formed in the first place. So this loss of 4 months, these memories were never consolidated. They're still sort of waiting around for that process. And the several months afterward, those memories just weren't formed. So how is this consolidation process working? There's a number of things that we think might be happening. One of them is long-term potentiation, and we'll start there. We'll kind of compare standard consolidation theory to multiple trace theory. I'm a multiple trace theory person myself, I think. I'm trying to remember which theory is which. And then we're going to talk a little bit about neurogenesis. Let's start with long-term potentiation. Essentially, this is the enduring facilitation of synaptic transmission. So, if you remember back to sort of basic principles of how neurons work, they work by generating action potentials. Well, long-term potentiation essentially changes the likelihood of an action potential occurring between two neurons by strengthening that connection. So we get this sort of long-term change in the electrical potential of a neuron, which means it's more likely to be fired. And so this is part of this basic cellular process involved in the creation of memories. We know this is disrupted by trauma, and we think this is also what's disrupted by drugs like midazolam, involving a neurotransmitter called GABA, because GABA actually reduces the likelihood of potentiation, and as a result, we don't get formation of memories. Yeah. Sorry, to the what? I'm sorry, to the what? Oh, the myelin sheath. Sorry, I keep hearing G. Sorry, I can't. Not necessarily. The myelin sheath speeds up transmission. This actually changes the resting potential of the neuron. So if you remember, it's at -70 or whatever, and it has to depolarize. It actually partially depolarizes the cell, so it's more likely to generate an action potential. So it has more to do with the polarization of the neuron. So remember, hyperpolarization would make an action potential less likely, and that's what happens with drugs like GABA and injury. So, yes, but no. How does that sound? So that's LTP. Again, this is an important process, but it's not the whole picture. Standard consolidation theory is the idea is that the neocortex is crucial for storage of fully consolidated memories, and that the hippocampus plays only a temporary role. So standard consolidation theory says that stuff just gets moved up into the cortex and the hippocampus is no longer involved. I think this is, I'm going to say this is Larry Squire's theory. So the standard consolidation theory provides that the neocortex is where everything's being stored. The hippocampus is involved in getting it into the neocortex, and not much more beyond that. I think this is kind of partially true. Certainly I think the neocortex is part of consolidation, but really think the hippocampus continues to play a role in memory long after memories are consolidated. And that's where we get to multiple trace theory. According to multiple trace theory, only semantic memories are entirely stored in the neocortex and are what we call hippocampally independent, because they don't require any reinstation of context. We think that episodic memories that have times and places and context associated with them, are dependent on the hippocampus. So that episodic memory continually relies on the hippocampus, and that new traces are created with each retrieval. So every time you retrieve a memory, you're reinforcing that connection. So this is this idea that we saw in the Memory Hackers video, that when you retrieve a memory, you're re-strengthening the connection or changing the connection. And so those episodic memories continually rely on the hippocampus for that. So when we talk about semantic memories, we're talking about them being independent of time and place and hippocampally independent, which is why patient HM still contains his knowledge, which is why Clive Warren still holds on to some of his knowledge, and also why patient HM was able to acquire some new semantic memories, although didn't remember any of the experiences associated with them, because it didn't require this hippocampus for that to happen. The mystery here is in the role of neurogenesis. I should also say, we think that genetic protein signaling is involved here, but I have no idea what's going on there, so I can't explain it. Basically, so every time you retrieve something, it re-strengthens the connection, so it's retracing the pathway. Yeah. So every time you retrieve something, it's sort of creating a new memory. So here's where some of the mystery lies. There's two places where mystery lies. Certainly neurogenesis is one of them. so none of these, neither multiple trace or standard consolidation or long-term potentiation takes into account neurogenesis, so the creation of new neurons. We think there is some evidence that new neurons may be involved in forgetting through restructuring. There's some people believe that the reason why we have infantile amnesia is because there's so much neuroregeneration going on that we don't remember anything from that time period. The idea that some people propose is that sort of new neurons are essentially remodeling the brain to form new memories, so it's sort of getting rid of old ones and forming new ones. That's really about as far as we can go. We also know that there are changes in the protein signals in the DNA of neurons that we think are involved in memory creation. We have exhausted my knowledge of that at the moment because it's so new and no one can really know. The field just doesn't have a handle on that yet. But it is leading to some very intriguing hypotheses that we might be able to inherit memories. I know, crazy. Because we know that epigenetic alterations in protein signaling are potentially inherited. So, for example, somebody who uses heroin, 2 generations from them, so their grandchildren and their great-grandchildren, you can still see epigenetic alterations in protein expression. This is a whole new area, so stay tuned for trying to figure out how all this works. So how does this all sort of work in terms of trying to establish some kind of model of how memory works? Well, we think the hippocampus and the entorhinal cortex communicate with one another, and then the entorhinal cortex, the parahippocampal communicates to the parahippocampal cortex up into the rest of the cortex, and then that comes back down to the pararhinal cortex. So this kind of reciprocal connection is one of the hallmarks of conscious memory formation. Because our conscious awareness is up here in the frontal lobe somewhere, we think, and so this being able to retrieve information back into the cortex is an important part of our conscious awareness of memories. Animal studies show semantic memories are associated with the pararhinal cortex, whereas context-dependent memories are dependent on the hippocampus. So this part of the memory formation, once things are up here into the entorhinal cortex, we're not having to rely on the hippocampus to restate that, or the pararhinal cortex, that's the wrong part. It starts to all get confusing. Semantic memories are up here in the pararhinal cortex. We can retrieve them without having to retrieve any context, because that's down here in the entorhinal cortex and the hippocampus. The temporal lobes then show asymmetry. That is, the right temporal lobes seem to be associated with nonverbal material that we can remember, and left temporal lobes with verbal material. No surprise there. Left hemisphere is long known for being locations for things like language. So the temporal lobes are certainly involved in storage of certain memories, abilities, that sort of thing. Verbal memory is more likely to be in the left hemisphere. Nonverbal, visual, spatial, that sort of thing on the right. The frontal lobes then are, of course, involved in working memory and executive functioning. We've talked about that already. But also in both encoding and retrieval. And so there's this thing we call the HERA model, which stands for Hemispheric Encoding Retrieval Asymmetry. You can see much greater activation in the left frontal lobes during encoding, and much greater activation in the right frontal lobes during retrieval. You can also see The greater their activation is in the left frontal lobe, the more likely something is to be retrieved later. And so one of the things when we talked the other day about effortful processing, the more you're involving your working memory, the more likely something is to be encoded, and there's certainly neuroscience evidence for that the greater your frontal lobes are involved in encoding, the more likely you are to retrieve something later. And then when you are retrieving something, you see that in the right hemisphere. A couple other things I want to quick note. Seems to be some difference in emotional memory. We think the amygdala mediates connections between the medial temporal lobes and other areas that help modulate this kind of emotional memory. We also think that connections with potential networks may be related here. So there's still a ton of work to be done in trying to understand how all of these pieces fit together. But the amygdala certainly is directly involved with the hypothalamus and the para-aqueductal gray, the medial temporal lobe and the basal ganglia. All of this probably leads a different kind of quality to emotional memories that we definitely want to consider. Finally, implicit memory. The reason why implicit memories are different is because we don't have reciprocal connections back to the cortex. So we have these subcortical structures that lead up to the cortex, but we don't have reciprocal connections back from the cortex. So it may be why these memories don't have a conscious component to them, because of that lack of a reciprocal connection. Parkinson's disease patients often have implicit memory deficits, and so we know that the substantia nigra is primarily involved in Parkinson's disease, something we haven't talked about in here much, but in my physio class we talk a great deal about. In Parkinson's disease, the substantia nigra is really decimated, and as a result we see Parkinson's disease patients have diminished implicit memories. And so we think that the basal ganglia and the substantia nigra are an important part of that. So the last couple notes I want to talk about are some applications of this general finding. First of all, cortisol is released during high stress and disrupts consolidation. So anything that's highly stressful is going to disrupt memory. Stress is just simply not good for your memory. So one of the things you have to do is engage in some self-care, trying to make sure your stress levels aren't too high, get some exercise, that sort of thing. Mindfulness meditation, which we talked about under attention, is another important part of that. So sleep's also a critical component. So taking care of yourself, getting a good night's rest, exercising, keeping your stress in check are all very important for really taking care of your memory and how it's going to function. So the last thing I want to talk about are some important emerging areas and tie a little bit of this all together. Right now the action's a lot of it's in neurogenesis. What's it doing? How's it involved in memory formation? How do we regulate it? Really interesting tie-in for me is related to depression. Depression patients have elevated cortisol levels. In fact, it's very clear that one of the things we know about depression is it's often triggered by a stress event. In fact, a lot of practitioners have started prescribing antidepressants to people who are going through a high stress event to simply try to kind of stave off some of this elevated cortisol levels. Depression patients also have reduced memory performance. This is kind of a thing that's been known for a very long time. One of the things we've also started to learn is that antidepressants, selective serotonin reuptake inhibitors, appear to induce or increase neurogenesis in the hippocampus. And so it's possible that one of the reasons SSRIs help some people with depression is because of this neurogenesis connection. It's speculative, but it makes sense. And so this may be an important part of trying to understand how those drugs work, because quite frankly, most people don't know how our Y-SSRIs actually treat depression. They just seem to. But they don't do that for everyone. It's also possible that's the reason why electroconvulsive therapy helps treat patients with depression. It's also why transcranial magnetic stimulation and transcranial direct cortical stimulation seem to be good targets for treating depression as well because of its potential role in neurogenesis. Final note, there are certainly sex steroids that are involved in neurogenesis. Estrogen seems to promote neurogenesis in the hippocampus, and we certainly know that progesterone promotes neurogenesis in the olfactory cortex. So this is another area in which we're going to start seeing some emerging research in how sex steroids affect the brain, in particular in this area of neurogenesis. Any questions? All right. Well, we will leave it there, and I will see you guys all on Monday.